Okay, welcome to this session, which is specific for chemicals. So if anybody's interested in biologicals, they should leave now. So anyway, welcome to this session, which is specifically on chemicals. The first topic is control strategy. The session is going to be led by Christopher Olofsson. He's a senior pharmaceutical assessor at the MPA in Sweden. Studied pharmacy at Uppsala and continued with a PhD in medicinal chemistry. I won't go into that. He's part of the CEP certificate suitability procedure as well at EduChem and an alternate member at the Quality Working Party. So I'll pass it over to Christopher then to lead this session. Thank you.
Okay, thank you, Keith. And welcome to this little more intimate session for the small chemicals. Yes, so I'm very glad to present Dr. Andrew Teasdale for our first presentation for this afternoon. Andrew is chair of Astra Zeneca's Impurity Advisory Group and head of external advocacy. He has been involved directly in the development of both ICH M7 and ICH Q3D and currently represents EFP on both Q3D and Q3C expert working groups. He is an author of over 40 papers relating to impurities, as well as 2 books relating to genotoxic impurities and ICH quality guidelines. Andrew is also the inventor of the purge factor concept used widely across the industry for the risk assessment of mutagenic impurities. So with this, I give the floor to Andrew.
Okay, thank you very much, Christopher, for the kind introduction. So if we could... Okay, so I think what we want to do is to try to maximize the amount of time that we have for discussion, because there was a lot of really interesting topics came up this morning, and some of the things that I will relate to, that I'll talk about, relate to some of the points that we came up with this morning. What I'm going to talk about is really an overview of some of the data challenges. All of the presentations this morning highlighted the fact that in a situation where we're doing expedited development, and one of the biggest challenges we have is the lack of data that we're able to collect meaningfully in the time that we spend as far as that development is concerned. And I'm going to be using primarily an example of an oncology drug that literally went from first patient in the clinic to first patient as an approved product within 27 months. So we had a very, very constrained timeline in that respect. And I'm going to look at it from the point of view of 2 specific aspects around impurities. There's the non-mutagenic impurities, where I'm going to talk about really primarily the challenges of setting a specification based on that limited data. And in particular, I'm going to look at this, how we ultimately may well be able to address that by closer alignment to the safety qualification data, and how that relates to the existing guidelines as well. What I want to try to do with a lot of this is try to really see what we can and can't use from the current regulatory framework. And actually, from an impurities point of view, there's a considerable amount of, how do I describe it, substrate or critical points within the existing guidelines that actually do allow us to, and are allied to being able to use an accelerated approach. I think that there is a good deal of flexibility and capability within the existing guidelines, and I'll look to emphasize this. And then I'll talk about mutagenic impurities, both in terms of ICH M7, but also as well the concept of ICH S9 as it relates to the patient populations, particularly in the context of what I'm talking about today, which is an oncology drug. You'll notice actually I haven't got anything on there about solvents and elemental impurities, and there's a good reason for that, because actually I don't see either of those 2 areas as a significant challenge when we're talking about accelerated development. It's really around the non-mutagenic impurities and the qualification and how we manage mutagenic impurities, I think, are the key areas really. So as far as non-mutagenic impurities are concerned, as most of you will be aware, the safety of non-clinical non-mutagenic impurities is established through that process of non-clinical safety evaluation, i.e. qualification. And it's based on the principles, as I'm sure most people are aware, of ICH Q3A and Q3B. But one of the challenges that we have with that is this whole chestnut, as we've talked about over and over again this morning, is how do you set a specification when you have limited data? And you clearly have that potential tension between the batch data and the safety data. And, you could talk about it being disruptive or whatever, but fact of the matter is that we have a limited amount of data from the point of view of understanding our process at that stage. And perhaps one of the guidelines where that kind of conflict really shows is in regards to ICH Q6A, which specifically directs that acceptance criteria should be based on, for instance, that whole idea and concept of mean plus 3 standard deviations, which actually in many ways, particularly if you're talking about a very small data set, you could really easily argue is not statistically significant. And also as well, one of the ironies is that if you're relatively tight in your development during that stage and you have 6-7 batches, that actually that's going to artificially constrain you at a very early point in this. And that's where you have that real challenge. And also as well, that challenge of relevancy as well. What batches do you bring in? Do you bring in some of the, the lab scale batches or the small scale batches? Or does it all have to be relevant batches from the point of view of manufacturing scale? And so that's one of the areas where there's a real challenge. And in regards to the specific case study that I'm talking about, this is one of the areas where the ability to be able to interact with regulatory authorities was of primary importance. We were able to have the dialogue primarily with the FDA now in our particular scenario, whereby we could actually articulate that this is the number of batches we had, this is what we know about this process at the moment, and also as well, this is what we know from a clinical point of view. And this whole point around, setting specifications and clinical relevance is one that's a very hot topic at the moment. The diagram I put on here is from a paper that ISPE have just recently published that looks at the whole issue of clinically relevant specifications. Now, what is articulated here is the kind of thing that you might apply to an assay, or maybe even something like a measurement like particle size. There you can relate it to a critical quality attribute and relate that through, for instance, to dissolution and actual clinical performance. But it's a little bit more difficult, particularly when you're talking about, as I'm primarily talking about here, about non-mutagenic impurities. For start, you don't have that lower specification. It's really the upper end that we're talking about here. And so just to put this into a little bit of context, I've taken, in a very simple way, some of the data from the project I'm talking about, just really to illustrate what was the impurity range that we saw. And this is less than 10 batches that we're talking about here of the API. And the impurity range, anywhere from not detected primarily of the primary impurity that we were concerned about, the one where we had to set a specific limit for. So it ranged anywhere between not detected and 0.23%. And if you took the mean plus 3 standard deviation, it gave us a potential specification by the time you round it down of 0.3%. What's the problem with that? Well, look at the bottom end of that range, 0.23, and if the limit was 0.3, that's on 7 or 8 batches. There's a very, very real risk that if we'd set that specification on that limited number of batches, based on a limited amount of data, that we could easily find ourselves in a situation very easily, as soon as we started in full-scale production, of actually finding that we started to fail batches. And now to put that into some context, that same impurity, when you look at the preclinical data, was qualified up to 10.3%. Now that's not typical, but normally what we do see is, in a number of cases, we do have that significant margin between what is safe from a preclinical point of view and the actual levels that we see in the routinely within the batches. Now, I'm not sat here, and nobody sat here saying, well, 10.3% is the specification. That's ludicrous. Clearly, that's absolutely the case. But what we tried to articulate in regards to our submission was that we should be able to bring that into some context. And what we agreed in this particular case was an acceptance criterion of 0.4%, which, to my mind, was still a little tight. We'd originally thought that 0.5% would have been a more appropriate level, but after some negotiation, that was agreed that that was the limit that we worked to. And that's proved in practice now to be an acceptable limit. So I guess the real point I'm trying to make here is that, we need to have some degree of flexibility that takes into consideration not just the process capability, but also as well, obviously, the preclinical safety as well. And really just to emphasize that a little bit more as well, and I'm no statistician, but talking to my colleagues within stats, one of the things they say is that, particularly if you look at, say, something like a specification for an assay, if you do that on the basis of the distribution that you see on on a limited data set for an accelerated product, that actually what you see is you see a process shift potentially of anything up to 1.5 sigma. And apparently that's fairly typical. And the consequence of that would be if you overly tighten that assay in that first instance, just because of the actual process drift and also the actual measurement itself, including the measurement variability, that all of a sudden you can find that will drift and you'll be out of specification for that reason as well. So again, it's a cautionary note about making sure that we don't over tighten specifications based on limited data. So that's what I would call normal impurities or non-mutagenic impurities. But if we now move to this, the circumstances around mutagenic impurities, now this is an example where we have a very, very effective framework already existing within the guideline. ICH-M7 provides a very, very effective framework for the development of and control of mutagenic impurities for an oncology drug. And there are two key concepts that are built into the guideline which we can take advantage of. The first is the limit can be based on a risk-benefit, and this is where it is aligned to the ICH S9 guideline around advanced cancer therapies, which takes into context the seriousness of the disease area, but also as well what the patient has been exposed to in terms of the treatments and the risk that's associated with that. And also as well, there is that clear alignment to the limits to duration, particularly to the Haber's law, and you have the table that's taken straight out of ICH M7 there. So that gives us the ability to be able to contextualize this, particularly in the context of an oncology drug. And so particularly really looking at that relationship with S9, what does the guideline say? Well, ICH M7, very clear now, it says that ICH M7 does not apply to drug substance or drug product intended for an S9 population. The only challenge with that is that when you look at what S9 says, is it pretty much sort of plays tennis a little bit with this and says, well, yes, you shouldn't set the limits as defined by M7, but it doesn't really tell you what those limits are. And that's where some degree of flexibility is required. The example I'm talking about here was this drug Tagrisso. In this particular case, what we took into consideration was that this was an advanced disease setting. The patients had already been treated with the previous therapy that had then become resistant to that before they were given Tagrisso. So we felt that very much met the criteria of ICH-S9. And they also had an expected life expectancy lifetime of less than 5 years. So we took the durational limit and the patient population into consideration. And so the acceptable intake that was accepted in this context for this oncology drug for a mutagenic impurity was 100 micrograms per day, which was considerably higher than certainly the standard TTC, and gave us that flexibility, particularly in regards to this medicine, where we had a relatively late mutagenic impurity in the synthesis, to exercise control at a level that was felt to be appropriate, and therefore meant that we could develop that process relatively quickly. Another key aspect of ICH M7 as well that relates directly to impurities is the flexibility that we have around control options. We have greater flexibility in terms of that mechanism to prove absence. Options other than to simply test, as we certainly did previously, for the presence in the final API. Now, it's not in the slide, but one of the kind of ironies of this is that the more reactive a mutagenic impurity is, the less likely it is to carry through to the API, but how much more difficult it is from an analytical point of view, just because of the difficulties in terms of analyzing things that are highly reactive at a very, very low level. So M7 recognized this and then built on that, and within that it has the control options in there, options 1, 2, 3, and 4, with the primary one being, and I've deliberately ordered this way, is option 4, which is it's so reactive that no testing is required either from an analytical point of view, and you can actually prove that that impurity is not present on the basis of principles defined by the purge factor calculations. And that's the basis, really, what this next slide describes. So this all aligns to what's captured within ICH M7, but is the purge calculations that I played around with 7-8 years ago now. Really what you're trying to do here is do what a chemist does in their head. You relate the physicochemical properties of a mutagenic impurity to the process it sees, that downstream process. Is it reactive? Is it soluble? Is it volatile? Or are you doing other operations like chromatography to remove it? And then what you do, and the trick with it is that we have a numerical score associated with it. You essentially score that risk. It's a numerical risk assessment process. And you simply take the physicochemical properties related to the process parameters, and then you decide whether or not you see an appropriate purge. And from that, you assign a numerical value to that. Then you multiply that up over the entire process. And ultimately what you're doing is a very simple mathematical calculation. You know what level of purging is required by the limit you've been able to set. You can calculate what the extent of the purging is from the purge calculations, and you simply look at the ratio. And that's how you can make decisions around what is an appropriate control option. And that's exactly what we did in the context of this particular examples. And that, just for further information, is just that scoring system. Just to point out and emphasize, it's a very, very simple scoring system. It always has been, and it will remain that way. You know, we decided we wanted it to be binary and easy to understand. It's either reactive or it's not. It's soluble. It's not. We didn't want it to be in any way trying to predict a purge of 625,000. It was like, is it 600,000? Is it 60,000? That was the whole purpose of this, to put that risk into some degree of context. And actually, as far as M7 is concerned, just to kind of show some symmetry on this, what you'll see is if you look at M7, not only does it describe the physicochemical properties, this whole idea and concept of comparing physicochemical properties to reactivity and using these purge calculations, it directly references a key paper that was written on this back in 2013, a cross-industry paper that showed the utility of this. So if I now relate it to that particular example, Tagrisso or osimertinib 9291, depending on which is the sort of, either using the internal or the brand name for it. And so what we did is we carried out a full-blown assessment from a mutagenicity point of view. We started, if you can analogize it, with a funnel where we looked at the types of impurities that may be associated with it, the raw materials, the registered start materials, the reagents, the intermediates, the side product reactions that we saw. And then what we did was the first part of it is the structural evaluation. That's really where you start that whole process off from a mutagenicity point of view, is does it predict for mutagenicity? Well, on the basis of that, for this particular molecule, we saw 10 potential impurities that fell into class 3, could be mutagenic. Of 3 of those, when they were subsequently tested, 3 of them were found to be Ames positive. Just to make the point there that when it comes to mutagenic impurities, it's pretty much impossible to avoid mutagenic impurities. It ultimately is really all about control when it comes to mutagenic impurities. And then what we did was, then we did the purge calculations. And the critical thing was, for an accelerated development, was that of the 10 of those impurities that were still of concern, 9 were found to be purged well below the TTC calculated for the drug. And so from that point of view, our control strategy for mutagenic impurities was massively simplified, because we only actually had to have one specified MI and one analytical method as opposed to 10. So that again was something that really helped us to accelerate development. And this was just an illustration of where those impurities came in. So there was an isolated intermediate, which was a class 2, came in with one of the starting materials, several at later stages. The key point is that the impurity that we had to ultimately control in the specification came in with the final intermediate, which is perhaps not surprising, because an awful lot of this does relate to proximity, and it may be expected we would have control over that anyway. But that was the one and only option 1 control we needed to exercise for mutagenic impurities. So the conclusions regards this, really, it's going back to the guidelines. You look at ICH-M7, and ICH-M7 is a very, very well-written guideline that actually, whether deliberate by design or not, does align very, very effectively with the concepts and principles that we want to articulate and see as far as accelerated development is concerned. You have those key concepts of limits based on duration and patient population, and that flexibility we have with control options. And also as well, but if you relate that then to normal impurities, this is perhaps where the more challenging area is that, you know, pragmatism is vital, and we perhaps do need to establish some well-established challenge some well-established concepts, particularly that within Q6A of mean plus 3 standard deviations. This is where I think ultimately we need to keep sight on the fact that we want to deliver that high-quality, safe medicine to patients, but we want to be able to deliver it. We don't want to find that, you know, we get a breakthrough status and then all of a sudden we're seeing OSs regarding a particular impurity because we've artificially constrained that limit for it. And really the key thing for me is that a lot of this can be gained through the dialogue. We managed to find a pathway forward, particularly on the non-mutagenic impurities, through the discussions that we could have because we had that breakthrough status for this drug. So these were the key points I wanted to get across, and hopefully that's given people food for thought and things that we can kind of now get into, hopefully, a discussion around. So thank you.
Yes, thank you, Andrew, and also for keeping time very well. As we got more time than we perhaps expected from the beginning, maybe I can pose the question if there are any direct questions to Andrew's presentations before we get to the panel discussion.
Hi, I'm Peter Kaspers from the Dutch Medicine Evaluation Board.
Hi.
Thank you for your very good presentation. I'm familiar with the purging work you and your colleagues have done, and I've read the article. But I have not so sharp how it is with the validation at this moment. Have you validated your system? I would say.
Yeah.
Also on several chemicals, because we regulators always want to see test results, of course.
Yeah.
And I think the ideal situation is that, like for quality QSR, that you can depend on your system. In this way, it would be nice that we could depend also on the system as being validated. Yeah.
Yeah, I mean, that's a really good question. And there's two aspects of the answer to that is that certainly when this was originally developed, it was a calculation carried out on paper or at best within Excel. And because of that, one of the things that we did, and certainly when I did the first publication on it, the first thing I did was look, well, does that number look sensible compared to the actual results that we get when we actually analytically measure it? And what we found, both in publications that I've been involved with directly, but in multiple other industry publications, is that when you compare the purge of an impurity to the predicted purge, that there is a systematic bias associated with this, and it's a deliberate one, that it actually underpredicts. And example after example across all of the published literature on this, and internal anecdotal evidence as well, is that it underpredicts typically by about a factor of 10. That whatever you see as a predicted value, when you measure it, it'll be tenfold higher typically, sometimes more than that. Why is that, and what's the systematic bias? Well, the bias relates primarily to solubility. We have a very conservative scale in terms of solubility. It ranges from 1 to 10. Now, we know in certain circumstances, in a recrystallization, in a purified solvent, with control over the recrystallization rate, where your impurity is structurally very different to your API, you can see significant purge way in excess of tenfold, and actually, in some cases, 1,000-fold. So we know that that's where the comfort comes from the point of view of knowing that we will always underpredict in that context over a process. Now, that's on paper, but what we're developing now is that we're now developing this as an in silico software system. To your point, really. So alongside LARSA, who are the scientific charity, we're now developing an actual software version of this, which we're now effectively rolling out, which is called Mirabilis. And within that, we can do far more around validation, because what we can do is we can build a knowledge base that actually makes the predictions for reactivity. So that system, it will tell you what the chemical transformation is, it will tell you what the impurity is, and it uses a knowledge base to then look at what evidence do we have to be able to say that in that scenario, if that type of impurity was present in that transformation, this is the purge we'd expect. And so we have that knowledge base that's been developed within that software. And that's completely transparent. If you had the system, you'd be able to see as exactly as I would what the reason is why a particular impurity was given, for instance, a reactivity value of 100. So the whole thing is transparent. And so you have that, literally all the way through that, you can trace it back to the scientific basis for that actual prediction. So that's really the evolution of the system, which I hope answers your question.
Mostly it does. I was just curious how many syntheses and processes you have actually used to validate the system. I'm speaking about hundreds or less.
If you look at it from the point of view what's in the public domain,
yes.
Then there probably are somewhere in the region of about 25 plus syntheses that have been assessed that have actually been published. Internally within organizations who themselves self-validated this to have the confidence to use it in regulatory submissions. Within Astra Zeneca alone, I've personally overseen something like in excess of 50 syntheses where we have some kind of comparison between predicted and actual measured. And it would be the same in many other organizations as well. I don't want to put anyone on the spot, but if Ron wanted to comment at all from a Pfizer point of view, I don't know.
Thank you, Andy. I can try. Like Andy, I also chair one of the company impurity councils, so I've been working with Andy in this field for a long time. His answer is very good. There's a lot of really good ingrained information now that says that this tool is, if you like, under-predicting the actual purge that you see. But from my perspective, one of the things I've always been very happy to see is that, of course, you don't just have to believe the tool. So if you get a predicted purge that's quite close to the purge you need to get to your control limit, get some more data. Validate it as you go. If you get a purge that's well more than you need to get to your theoretical level, trust it. So it can allow you to kind of make risk-based decisions on where you get additional data as well.
Yeah. Oh.
And for me, that's really helpful to the development teams. And it would appear really helpful to the regulators who want to understand how the tool is used.
Yeah.
I'm hoping.
Yeah. If I could literally draw your attention to this as well, very, very quickly on that point that Ron had just brought up. We've actually written a paper now which looks at how do you contextualize this. And it's exactly to the point as Ron has described. A lot depends on how close the ratio is. And the closer the ratio is to what is required, the more analytical data that you would create. And actually this paper has been published at the end of last year, and it's all done predicated on the basis of the purge ratio and whether or not it supports an option 4 approach. And really the key thing is that when the purge ratio is greater than 1,000, that's when we say that we're confident of the predictions with this system. No further data is required, particularly considering the conservative nature of it already. But if you look at it from the point of view of if the actual I haven't got it in there. But if you looked at it from the point of view, if the ratio was, say, between 100 and 1,000, in that case, then we would collect some more confirmatory data. And if the ratio was less than 100, then in that case, then we would say and have stated as an industry that we believe that if it's less than 100, then we would expect to get confirmatory data, maybe spiking purge data, and also as well, maybe even batch data as well. And if in that case we still didn't have the confidence, then you go for one of the other control options, primarily option 1, which would be specified on the API specification. So, yeah, we really thought about that kind of question you've asked, and we try to sort of standardize in a way by making a decision process around that link to that ratio.
Thank you for your response. Yeah.
Okay.
Thanks. It's Matt Bobkin, GSK. So I don't run the Impurities Council at GSK, but I sit next to the guy who does. So just to echo from a GSK's point of view, Andy's points and Ron's points, and the fact that, I think the models are being, and I use the term model because it's a type of model, are being thoroughly verified by the industry efforts that Andy is leading. I wanted to reflect that it's a prior knowledge case, a classic prior knowledge case, where what is known collectively across companies and the experience we have is greater than the sum of what any one company has or any one organisation has. And as a classic use of prior knowledge, there's obviously a benefit in turning that prior knowledge into established knowledge, which is why I think the publication strategy is so important for everyone on either side of this room to actually get to the same level of understanding around the trust that you can put in them in these important calculations of molecules. So, in that context, I think it is a very interesting example of how prior knowledge can accelerate development. And my build is, of course, that not relying on wet chemistry assessment, i.e. not relying on experimentation to test hypothetical impurities, gives you the opportunity to explore impurity space based on consideration of what could be in there more than you've just seen. So it's actually a better scientific approach and gives you more benefit in terms of what you can assess, and therefore a greater opportunity to address risks than the limited classical chemical analysis approach that we've historically taken.
And just building very, very quickly on that, I mean, as many of you will be aware, we've had the incident recently with valsartan, the contamination with nitrosamines. We have a drug ourselves, which is sartan, which is candesartan. And actually, as soon as the issue hit, we tried to work out how it could have been formed, what were the reagents involved. And one of the first things we did was a purge calculation to calculate, could we have any actual risk? And within half an hour, we calculated that actually for our manufacturing process, that actually there was no risk of generating NDMA. And subsequently, regulatory authorities have asked for further data, but actually to totally reinforce what we got from the purge calculations. But that allowed us very, very quickly to be able to ascertain whether or not we had a risk. And if we had a risk, then we could have reacted extremely quickly, far, far quicker than the several weeks it took us to develop a method for a nitrosamine. So in that context, it gives us the ability to be able to react very rapidly to potential challenges as well, not just about avoidance of analytical testing, really.
So thank you. With the discussions now well underway, I think we're ready to the panel discussion, which might just be a continuation of this, but we'll see. So I'd like to take the opportunity to present the panel. I think myself and the speaker and two of the FJ members, Mahesh Ramanathan and Ramesh Sood, have already been duly presented. But I'd like to also introduce Scott Furness, who is the Deputy Director of the Office of New Drug Products within OPQ-CDR. His expertise in synthetic chemistry and regular review topics associated with synthetic and semi-synthetic drug substances. And just to aid the discussions, if we would need some help, we've had some topics for those of you who can see the slides. So some of the topics we raised was the use and acceptability of in silico methods, which we've already been touching upon, and also the applications options 3 and 4 of SHM7, which I think also Andrew outlined. So the floor is free.
Christopher, can I be bold? I actually think that the ICHM7 guidance is giving really strong and clear advice to people now. And it's not the big issue in the field at the minute. It's dealing with a significant potential hazard, but I don't think it's the biggest risk or issue in the field. I don't want us to forget what Andy was saying about the specification setting on the basis of limited batch data. I think that is actually one of the big challenges, because industry can find that with a qualification understanding, then the specifications can be challenged in different ways by nearly every country that you take it to. Now, for me, that's quite an interesting position to find yourself in, not just in accelerated development programs, but more generally. So I think really one of the really intriguing pieces is, can we get to a position of a little bit more allowance and acceptance for Andy's type of approach, where it's not based strictly on the actual batch data you've got, but in the context of the other understanding you've got. For me, that's really important. I can see some agencies publicly beginning to look and support that kind of approach. And actually, I know that in other quarters, you can get considerable pushback on it. And for me, that's currently one of my big challenges internally, because my teams really don't know when they're going to have a specification agreed and when they're not. I wonder if there's any views on that.
I got one very, very quick build, which is to your point about prior knowledge. There's a considerable amount of prior knowledge that exists now, and some work that was done by FPA that I was involved with, which was led by Jim Harvey at GSK, where we managed to find substantive data to really underpin the 1 mg a day value that's defined within ICH Q3A. That would be extremely valuable if we could get acceptance of that validation of 1 mg, because often what we find ourselves in even in some extreme circumstances of a qualification level could be higher than the ICHM7 TTC under situations where you have a very low dose, and scientifically that doesn't make any real sense really. And so I think, you know, to Ron's point, I don't know whether this is the forum for it here, but I think it'd be really good to be able to have that further dialogue around whether or not Q3A ultimately could be underpinned by that actual limit of 1 mg a day rather than the percentage, which I think if you talk to many toxicologists, say, well, percentages in the context of, of safety is not really a relevant approach to take. But so I'd just put that in there for the debate as well.
Yes, thank you. Jobst?
Yeah, I just have one question. So if you do your purging calculations, you look only at the synthesis itself. So you cannot manage any mutagenic impurity which is imported by solvents, reagents, or whatever.
No, you would. No,
Because I think from the systematic...
No, you would.
you can-
No, as part of the risk assessment, if you look at regarding the wording of ICH M7, I mean, it does talk about the need to consider probable and likely side reactions, degradants as well, and things that could come in with, for instance, with an RSM. No, I mean, I think I can speak fairly clearly for most in the industry, because there's a lot of discussion we have around this and have had for best part of a decade. We do make a conscious effort to understand if there are specific mutagenic impurities could come in from, for instance, a side reaction. So I'll give you a good example. If you bubble HCl gas through methanol, you will generate methyl chloride. You'll generate about 10% of that, but that's a very low potency carcinogen. It's highly volatile and very soluble. So you generate it, you can purge it, but it has to be part of your risk assessment, and you would look to articulate that. So every effort is made to understand whether or not mutagenic impurities could come in, certainly from starter materials, certainly from reagents as well. I mean, there's some particular protecting blocks like something called Semchlor. I don't want to get too much in the chemistry, but it's pretty well known that if you use that particular protecting block, it contains bischloromethyl ether. So there's a lot of knowledge that we have around this, around reagents and things like that could generate a mutagenic impurity, or could be low-level impurities present in common reagents that we use. Now, I think the one trick we need to do, particularly after this issue with the nitrosamines, is from an industry point of view, more clearly articulate what we do know and actually what we do take into consideration in a way of better answering your question.
I would say part of it. So I would be willing to trust such a theoretical in silico concept at the time of marketing authorization. But I wonder what happens 10 years later, because you do your predictions, you have your prerequisites, and then you change some sources of starting materials, you change some solvents, whatever, and do you plan to do such a systematic evaluation each time you do a validation? Because normally then it's licensed product, it's on the market. Now you do your changes and you just make I would call it little risk assessment and say, okay, that's fine. And therefore, from a very conservative point of view, it would be very nice to have some changes underlined by some representative batch results or something like that. And I'm a little bit reluctant to trust this theoretic calculation for the next 100 years, just to say it.
Yeah.
So is there any possibility just imagine you would be a regulator to say, okay, this strategy is based on theoretical calculations, and each time a major variation will happen, this should be re-evaluated.
I think that goes without saying that that's what should happen. Yes, absolutely. I totally support that. I know that certainly as part of our change control process that we have around all of our established products, that's entirely ingrained within our written procedures internally, that if you make changes to your process, particularly, for instance, certainly a new route would trigger it. We would have to do a full-blown assessment of that. But the expectation is that even if we're talking about changing a solvent or something like that, then that would at least go through a preliminary assessment to see whether or not that could either change the actual basis and argument of the actual purge argument, but also as well whether or not it could introduce a new impurity. And I mean, the classic example of this we have now with valsartan, where changes were made without really thinking about what the consequences are. Now, most chemists would know, looking at if you take DMF under acidic conditions and you put sodium nitrite there, you've got a risk that you're going to generate nitrosamines. It's established chemistry, but that wasn't picked up in that change, and that change wasn't documented. So I think we should be doing it already, but I think we've absolutely had a salutary warning from that valsartan situation that says that absolutely you must do that. So I totally agree with every point you've made on that, and I think that should be part of everybody's process.
So Andy, if I could just refer to Job's comment from a regulatory perspective, representing my company, I can absolutely assure you that the same rigour and the same risk assessment is applied to post-approval changes as we apply to MAAs. That is beaten into our PQS.
Yeah, and it's not just one company. I couldn't tell you how many companies are doing this. But it's really important if industry are talking to you about a life cycle approach to managing quality, that they have got a really strong view on what change management has to be. Now, then the question is that something that gets inspected? Is it something that gets assessed? And to be honest with you, we would be happy to share that kind of question and an answer to it if you have concerns about it. But like Andy, we too would be building that into change management and variations for process changes.
I think Jean-Louis has asked for the word.
Yes, thanks, Andrew, for this excellent presentation. First of all, just to clarify also what Jobs has said, basically each change has to be, this is part of the quality system. So you have to make an evaluation. This is also part in ICH Q12 where this is indicated. And you have a nice table where you have to evaluate what's the implication of the change and so on. But basically, what you described here is really complementary with Q11, because they also speak about purging, about and so on. So, and as Ron was in as was in Q11, so I assume that he got passed the message. But basically, what you have presented here is independent of our discussion of today of prime. This can be, this is something I expect you would do for any type of application, independent if it has to be an accelerated or not. Would we expect, or would regulators expect, when you present your model and so on, that you, especially in a prime situation or an accelerated situation, as a commitment, would we submit then additional data to confirm your model, or would you say, no, our model is so robust that we do not need to submit any confirming data?
I think it goes back to some of the discussions we've had that we would be, on the basis of those principles that we published from an industry perspective, if the ratio was predicted to be over 1,000 between predicted and required, then in that case we would argue and contend that irrespective of whether this was a prime application or a normal development, that would be acceptable. I think if it was certainly the case where we had a purge ratio that was less than 100, all we had was spiking and purging data and limited batch data, then that's a scenario where you could argue that there could be an argument for saying that as part of a commitment post-approval that we would commit to actually generate more batch data with a view maybe even to as a sunset clause, remove that specification for that particular mutagenic impurity in the longer term when we have more assurance from more batches. So I think it depends on that ratio, but certainly I think in the case where the ratio is less than 100 and we have limited data, then definitely I think we would see some requirement there for some post-approval commitment to actually generate more data to support our arguments, particularly if you wanted to remove that test in the long term.
And all these purging processes would run under GMP. This was my contribution to the discussion on API starting material. Sometimes we have these issues that for the API starting material, we know that upfront some mutagenic. Then it's the question, should they be part of GMP or not? But forget this. It was just a nasty contribution.
Well, we enjoyed that contribution on the Q11 working group, Jean-Louis. And to be honest, the connectivity between these working groups is really important. It wasn't me who carried that message forwards. It was Steve Miller, who was both on the M7 team and then on the Q11 team. So there's a real connectivity with some of these things. You can't shake your head at Steve Miller, please.
I just wanted to be once nice with you.
That's fine. And I'll be nice too. Because to be honest with you, your question about is this something that we do only for prime or something more general? These approaches can be more general. Yeah.
I accept that, but that was my question.
So my rejoinder to that is that in a prime approach, when you've got very limited batch data, the piece that Andy's talking about not being tightly constrained to batch data is really important, because you can find yourself managing in the small molecule space what vaccines we're talking about this morning, out of specification results that may have no true impact to quality or safety or efficacy to the patient. And actually, that's something to try and avoid if we can. And allowing it to be based on a more qualification-driven approach, rather than immediately on the basis of batch data, is really helpful, really valuable.
So I would like to thank Andrew and all participants in this discussion. I think we've used all our precious seconds for this discussion. Thank you.
Thanks, Christopher. Before we leave the subject, can I just ask a question? You made the point about Q3A and changing percentage to 1 milligram. Can I ask, is industry actively pursuing that at the moment?
Yes. So, I mean, what about over 2-3 years ago, we had a FPA meeting where we talked about a variety of different things that relate to qualification. One was the principle and the basis of the 1 mg. Could it be defended scientifically? We also looked at the issue of impurity qualification during clinical development. Could we apply some kind of modified Haber's law to that approach? And there was a paper that was ultimately published on that last year. Now, what's happened since is that there's continued to be some discussion with that. Some of the discussion has actually shifted from Europe into the US. There is a Winter Tox Summit in January where there is a session where this is going to be discussed in more detail, and it'll cover things like the 1 mg. It'll cover qualification based on durational limits and the other chestnut, which is mg per kg versus mg per meter squared, which seems to come up just about every other year as far as qualification is concerned. So there is another really important discussion coming up on that at the beginning of January.
Okay, thanks for that clarification.